Our next speaker is Dr. Johannes Von Lintig. He's the professor of pharmacology at Case Western Reserve University. And we're going to change gears a little bit, and he's going to talk about carotenoids, colorful pigments for sight and life. Johannes.
Thank you. First of all, I want to thank the organizers for putting together this nice mini symposium. My name is Johannes Von Lintig, and I'm a professor of pharmacology here at Case. My research lies at the intersection between pharmacology and nutrition and deals with nutraceuticals. The nutraceuticals of my choice are the carotenoids. You all know those pigments as red to orange colors from nature, and they play important functions. Beta-carotene, which you know as the orange of carrots, is the major precursor of vitamin A and is converted in our body to two molecules, 11-cis-retinol and all-trans-retinoic acid. And through these molecules, beta-carotene affects vision, cellular differentiation, reproduction, metabolic control, and embryogenic development and organogenesis. However, we do not only absorb provitamin A's, we also absorb other carotenoids which have no provitamin A activity, such as lutein and zeaxanthin. Those pigments serve as light filters and antioxidants in our tissues. The highest concentration of those compounds you can find in the yellow spots, the maculae of our retina. And these macular pigments are responsible to reduce chromatic aberration and increase visual acuity. My lab has over 20 years dealt with this metabolism, identified several genes, and characterized their function. This is a picture of what we have currently about this metabolism. The most important thing with provitamin A and retinoid metabolism is that it is associated to many diseases which are depicted here. Today, I do not want to talk about the metabolism as a whole, but I want to talk on an unanswered question: what is the mechanism behind carotenoid accumulation in the human macula? And to do this, I have to introduce the first player in the scheme, yes. And that's the enzyme beta-carotene oxygenase 2. Beta-oxygenase 2 is a mitochondrial enzyme and is a ferrin-iron oxygenase, which basically breaks down carotenoids such as zeaxanthin into smaller entities. It's expressed at the inner membrane of the mitochondria. Mice normally do not accumulate carotenoids, but when we knock out, by genetic means, BCO2 gene, we have the following pictures. When we feed now these mice with carotenoids, they turn yellow as corn, golden as marigold flowers, or in the case of astaxanthin, red like salmon. This enzyme also plays an important role in nature. It colors the world around us. We showed together with Andres Bedensky from Columbia University that it's responsible for the color polymorphism in our pet fish, such as betta fish. Together with Michael Brown and his co-workers at Smithsonian, we showed, for example, that white and yellow, golden colored mannequins are differing by mutation in the BCO2 gene, yes. In the region in Central America where those species overlap, the yellow variety is the most common one. The reason for this is simple. Females prefer the yellow males and therefore the BCO2 gene which is coming with it. When we are now translating this into the human eyes, you would expect that BCO2 expression is high in the peripheral retina where no carotenoids are and low in the central fovea where carotenoids accumulate. If you now look and do single cell sequencing, you exactly find this, yes. BCO2 is one of the most preferentially expressed genes in our retina and is highly expressed in the periphery and lowly expressed in the BCO2 in the central part where the carotenoids accumulate. And we confirmed this here with help of Iverside and Onkar Savant, who provided us with retinal samples, and we showed this here by RT-PCR analysis and also Western blot analysis. If you now look to photoreceptors, they are something which is relatively surprising. They are rich in mitochondria, and this is thus far surprising because they are doing glycolysis and do not need the mitochondria for respiration. According to our hypothesis, our research, one would now or could now conclude that they are rich in mitochondria because in the periphery, here in the mitochondria, the BCO2 dependent carotenoid metabolism takes place, and in the central retina, carotenoids accumulate in the mitochondria. However, the big question is now, how do the carotenoids enter to the mitochondria? That's not a trivial question because Carotenoids are lipids. They cannot simply diffuse in an aqueous environment like a sugar molecule or an amino acid. Therefore, you need specific transport proteins. And we looked for transport proteins which are expressed in photoreceptors. And we found one of the most highest expressed protein is Aster B. Aster B was recently characterized in the context of cholesterol metabolism and transports cholesterol to mitochondria for steroid hormone production. We then wondered, is Aster B really expressed in the retina? And again, with help of Eversight, we analyzed eye samples. You see that Aster B, which helps carotenoids to go to the mitochondria, and BCO2, which catabolizes the carotenoid, showing somehow opposite expression patterns. We also wanted to have spatial resolution, and we did immunostaining for this protein. What is here shown in red is the expression of Aster B in the retina. And if you're now zooming in the fovea, you see Aster B is expressed in the inner segments and also the outer segments of the photoreceptors. Aster B proteins have a domain-like structure. They are anchored within their C-terminal domain as a transmembrane domain in the endoplasmatic reticulum. They have a binding domain for lipids and then a gram domain which connects them to another membrane. Aster proteins work like bridges. They simply catalyze the movement of lipids such as cholesterol from one membrane to the other. To demonstrate now that the Aster proteins do the same thing with carotenoids, we invented a new test system. For this, we used a bacterial strain, and this bacterial strain we genetically equipped with genes for carotenoid synthesis. They are now producing carotenoids in their membrane. Then we express the Aster proteins. And the Aster proteins, if they really can bind carotenoids, we expected that they will extract them from the membrane and form carotenoprotein complexes. And that's exactly what we found. We already see here is this yellow color that the protein alloys of the Aster proteins, when expressed in this bacterial strain, are yellowish. Yes, here's you can compare it to the control. Also, the spectral characteristics tell us that they bound carotenoids. And molecular modeling now showed that a carotenoid, this really bulky molecule, perfectly fits into the binding cavity of Aster proteins. Thus, we conclude that BCO2, which catabolizes carotenoids and is expressed in the peripheral retina, and Aster protein, which transports carotenoids, are really important players in carotenoid metabolism in the macula. Here are our future direction. People who are suffering from mutation in Aster B protein display recessive neurodevelopmental disorders with intellectual disability. For this, we have to say Aster protein for sure transport other lipids too, yes, not only carotenoids, but carotenoids. The human brain is really full of carotenoids, and it's thought that they have many functions as vitamin A precursors, but also as molecules which are protecting the brain from oxidative stress. And we are certainly now very interested in what eye phenotypes those patients may have. Thanks to our knowledge about carotenoid metabolism, we are also able to produce a mouse. Mice normally do not have carotenoids, which accumulates now carotenoids in the retina, as here shown by this chemical analysis. With this macular pigment mouse, we want to see whether carotenoids indeed protect the retina against damage from light and other abiotic stressors. At the end, I want to thank my people who were involved and who did all this research, and certainly also our collaborators. I want to thank here especially Onka Savan from Eversight, who provided us with retinal samples from humans. And I'm happy to take questions, and thank you for your attention.